I'm going to discuss polymorphisms of methylene tetrahydrofolate reductase. Genetic polymorphisms of the MTHFR gene lead to a varying degree of reduced enzymatic activity of methylene tetrahydrofolate reductase. The degree of activity is dependent on which SNP, single nucleotide polymorphism, is affected. Two of the more commonly expressed variants are A1298C and C677T. MTHFR is a rate-limiting enzyme involved in catalyzing the conversion of 5,10-methylene tetrahydrofolate to 5-methyl tetrahydrofolate. 5-methyl tetrahydrofolate is the major methyl donor for the conversion of homocysteine to methionine. Methionine unites with adenosine to form SAM, which methylates DNA. Methionine is also used to make proteins, utilize antioxidants, and to assist your liver to process fat. This visual represents pathways for properly functioning methylene tetrahydrofolate reductase. You can see where it's involved in the conversion of folate to its more usable format, as well as the other reactions and processes that are dependent on that conversion or affected by that conversion. In an individual with reduced function of MTHFR, homocysteine can build up, since 5-methyl tetrahydrofolate is used to convert homocysteine to methionine. High levels of homocysteine have been linked to increased risk of heart disease, stroke, Alzheimer's disease, and osteoporosis. Deficiencies in MTHFR activity lead to elevated levels of homocysteine in the blood. DNA methylation is also affected. A deficiency of methyl groups can lead to reduced methylation, and reduced methylation means that some of the DNA transcription and regulation is lost, which, as you can imagine, is not good news. So, big picture potential effects of impaired methylation include gene expression, cell differentiation, and chromosomal instability. Those can then cause a wide array of effects and expressions in an individual. We may see it as cancer, lupus, muscular dystrophy. A range of birth defects have also been linked to defective imprinting mechanisms. MTHFR polymorphisms are genetically inherited, so you either have it or you don't. However, there are a couple of dietary factors that can exacerbate the negative effects. For instance, folate status. In individuals with the MTHFR 677CT polymorphism, they are especially affected by low folate, and it's associated with an increase in homocysteine concentration and DNA hypomethylation. Alcohol consumption is also a factor. Alcohol has negative effects on folate, including malabsorption and increased excretion. Alcohol consumption and correlation of increased cancer risk may be related to alcohol's role as a folate antagonist and its suppressive effects on methyl group metabolism. Those individuals with the TT genotype of MTHFR 677CT are especially sensitive to these carcinogenic effects of alcohol. So for those individuals with MTHFR polymorphisms, it is pretty important that they undergo genetic testing to determine exactly which variation they have. Their treatment will be quite different depending on that factor. So that is all I have today. Thank you.